I'll just begin with what Mr. Dave mentioned, that sometimes you have a chemical. You have some idea that it could be risky, but you do not know the scientific data. And that was a chemical plasticizer, di-2-ethyl hexyl phthalate, on which I worked, which can leach out from the plastics. There were reports that in the blood, in the fluids, which pass through the plastic pouches, it goes there. And in 1973, Lord Tepper, that then FDA commissioner in the US, said that this is a chemical looking for a disease. Now, more than 20 years down the lane, it has been linked with a variety of the effects which you can give in the carcinogenic effect of this di-2-ethyl hexyl phthalate, as we discover that it has endocrine disrupting properties and can get along with many, many ways. So this is how a toxicology has to evolve. It has evolved, it keeps on evolving, and it has to evolve further when we come up with genetically modified products, etc. How you carry out the toxicology of that? How can you make an animal to eat more than 2 grams of feed when you have to feed them much more than that? So you have to evolve and bring in more. So this is points which I would like to touch in this presentation about the emerging toxicological issues. We have with us Dr. Rao, who has spent whole lifetime in that, in the toxicology, and I'm sure in the food toxicology session, much more these aspects will be discussed. Well, there's on the food safety, large demands are there, and you all are aware of it, so I'm not going to spend time on that. Foodborne illnesses, I'm sure everybody is aware of the figures, which we have, how many cases we get a year, and according to the WHO, 70% of the 2 million deaths per year from diarrhea in developing countries, as mentioned by Mr. Panandika. Hazards, biological, chemical, physical, is you face, which is and one has to be ready with the toxicological tools to carrying out the assessment of the hazards which they are posing. So you need microbiological food safety, residue, toxicology, and that can give to the safety. Still, it's a question mark that it will really lead to the safety, even after knowing, assessing them. Complex system, just mentioned earlier, numbers of steps where you can get contamination, and right from the farm to the fork. So one has to remember the contamination can take place at any of these steps. So the type of the challenges, emerging microbial threat, newer and newer coming, chemical threats, physical threats, and adulteration threat. You all know melamine story with the China came out. I'm not going to again read down all the names of these emerging foodborne bacteria. These will be discussed in detail. Like with this, the emerging foodborne viruses, hepatitis A and E, norovirus, avian influenza, which will be discussed again in you have a special session on microbial risk assessment. Foodborne parasites, number of them. The pathogens, again, these will be discussed, and I think Nimish is going to talk about these. So toxicology assessment, identify and characterize any potential adverse health effects. That's what the job of the toxicologist is. Inform and help prevent adverse health effects based on that. I think there's in the risk, there should be a predictive risk also. Risk assessment, a predictive risk. How can you predict the risks? Well, extrapolate toxicity from animals to humans, and I think we have to come out, and a lot of efforts is being done when you have to not to use the animals. More and more pressure is not to use the animals, but you have to come out with the models which can be validated and accepted by the regulatory agencies. And the whole new science is coming to the regulatory science, and again the FDA at NCTR is carrying forward this. And I attended a meeting in China where we had the whole symposium of 2 days on the regulatory science helping the toxicology. General approach, take up various toxicology endpoints of concern, which could be systemic, reproductive, developmental, genotoxicity, etc., with an aim for establishing an acceptable daily intake. Calculate the safe concentration for each edible tissue or the product based on the current food consumption values. The alternative approach provides an equivalent assurance of safety, which will be scientific justification, not to perform all standard toxicological studies or conduct focused toxicological studies. There's been more and more pressure because of the point that the costs which are coming up in the evaluation. Risk-based margin of exposure, weight of evidence, body of evidence, and justification based on the literature provide all that information. That's the kind of the alternative approach, but yet we have to convince the regulators, we have to educate the regulators, and make sure that these are the one which, again, make sure that they are not going to cause any toxicity. Number of, as I already mentioned, so this can be also help on the focused toxicological studies. We decide which is the remaining part which must be done and carry out those. What's future challenges we have? Traditional standard toxicology package may not be adequate for addressing the health, the food safety issues. There's concerns from novel products, nanoscale materials, more and more. These are getting in the food industry. Synthetic protein, hormone products. You know, we're talking of the nutrition. Now it is becoming nutraceuticals. People are bringing up and making the claims, but still somebody says it's a health supplement, food supplement. It will give you these beneficial effects, but never goes into testing the rigorous when you claim for a cure or for effects. Then the combination of the drugs. Nanosensors in pathogen detection, nano drops in oil, healthier oil. You know, when they advertise, I don't know what type really they have, nano or not, but they claim that they have nano drops. Silver nano and nano composites in packaging, there is no hard evidence that nanomaterials in products in the market will harm humans or the environment, but I think there is enough evidence to say that we need to examine, re-examine. But when it comes, are we prepared? Do we have the tools in toxicology to validate or assess the toxicity of these nanomaterials? These have unique properties. Absorption, distribution, metabolism, excretion may be essential, but how you do that? Case-by-case approach may be adopted, and this is how different agencies are working. Same with the synthetic protein and hormone products. Challenge, address oral bioavailability, I just mentioned that. Can we carry out in vitro, in vivo toxicology studies? Digestibility by GI tract of target animals or humans, and receptor-based metabolism of action and human relevance framework, exposure concentration. These could be, again, the avenues, opportunities to carry out. There are a lot of new emerging technologies. Computational biology, people say use genomic, proteomic, metabolomic. Yes, they can give the answers, but how much you can today utilize, and more, and this is one of the area which was discussed heavily in this regulatory science, the workshop which we had, and newer and newer test systems are coming up, so there's more on the research-based assessments. Another one, the biotechnology-derived food and products, approved versus unapproved, the limits of the detection methods, and what is the limit, how can you detect it, toxicity and allergenicity data, labeling requirement. So these are, again, the issues which needs to be considered, and this is the challenges they are posing. Safety of botanicals in traditional food. I'm not going to go into the detail of this, but basically, as I mentioned, supplements are food containing high levels of nutrients. So this could be the result that they can occur because destruction of nutrients, reduction of availability of the nutrients, interference with utilization of nutrients, and decrease in the food. They may be nutrient, but how a toxicologist can come up with the answers to the food safety issues and answer that. Again, there's a number of points which I'm not going to read out, all of them. There are claims that they, as I mentioned, for the nutraceuticals. So here the botanical traditional food. So this is again a concerning area. The other areas which I mentioned was the plasticizer, the food contact with the substances. Packaging innovation to ensure food safety as certain components of packaging like printing, inks, labels, colors, seals. You know, about 10, 15 years back, we were at a meeting. The lunch was brought from Narula, and the napkin, paper napkin, when it was just opened up, on the rice, beautifully, the imprinting came of that organization. That's the ink. So this is the, again, challenges, how you do that. Do we need innovative packaging and active packaging? These are all, yes, we may do, but we have to ensure that whatever changes we are making in the packaging material, that will not lead to another contaminant. Provides way to monitor and rely information regarding the status of the content, verifies information. The toxicity testing of the food packaging material have to be done in animal as well as human data are rarely available, and I don't think it can be available. I took that example of that plasticizer. When I was carrying out research, that is back in 73, 74, many people asked me, you are talking of something about so much of the plasticizer is given, animal doesn't die. What's happening? We were the first one to report its reproductive toxic effects, and later on, other people confirmed it. Glass, ceramic, cans, safety assessment of food packaging material. Again, this is all are the one has to concern what type of the material you're using for the food packaging. Today, the bringing out the fancy packaging material in the food industry for the making it attractive is all very important, but at the same time, they pose the challenges how you assess that safety. Chemical contamination, majority of them heavy metals. The story, everybody is familiar with the argemone oil adulteration. Happened about a few years back in Delhi. Thousands of the tankers were staying there. We didn't have the method how to find out the argemone is there. There was pressure that allow this tanker to go into the market, and again, this institute, ITRC, I was there, now it has a new name. From a center, it has become an institute, Indian Institute of Toxicology Research, IITR. We were forced that whether we should tell how much amount of the argemone will not be toxic. Because they found some study carried out in another ICMR institute, which said that in monkeys, this much of the argemone, if it is there, it is safe. But I quoted at that time the Food Adulteration Act, which said it should not be present. Nothing can be adulterated in the oil. And that's how we stick to that. And my colleague, Dr. Khanna, had at that time built a technology which was still the bench scale. That same was brought here and tested, and then the tankers were allowed. So these are the challenges, with the chemicals you get, with the material, microbial you get, and you have to face them. Genetic, epigenetic differences in population and different responses in food and food ingredients. I think it just suffices to mention the top lines, which will carry out all the messages. And as the CSR DG has worked out the genetic map of India, CSR is celebrating its 70th year foundation today. And this is the gene map that shows the differences in the people in Gujarat, people in Punjab, and people in West Bengal, how they respond to drugs. So this could be their responses to the food toxicity, material toxicity could be different. I'll skip this, nutrient-drug interaction. There are a variety of them, and these effects are being done. How you assess this? Do you have a protocol for it? Do we develop a protocol for it? I think this is again an area where people need to sit down and think and talk about it. So the concern, detection of environmental contaminants in water, soil, biota. What could be the possible environmental behavior which may be different for traditional substances, properties, entry into the environment? biologically active molecules and potential subtle impact. Little note about the impacts. And some contaminants may move from low to high risk due to land use change, influence of the climate change, which was mentioned by Mr. Dave. And this again is a concern, but we still do not have answers to them. Food safety challenges. Do we know all type of pathogens and chemicals causing food safety hazards? The identification of emerging food hazards and contaminants. Development, validation, application, innovative methods for detection and control of pathogens and chemical responsible for foodborne illnesses. Risk assessment and risk management in food safety. What is the most cost-effective point to intervene in the food supply chain? These are all very challenging as we deal with the moving targets. Pathogens are living organisms. They react to change environments. Mutation occurs in these organisms on a regular basis. Do we have toxicological tools to detect these mutagens or predict even could be the possible, and then what circumstances they will change? So large and diverse group of chemicals, new analytical methods required, behavior different from traditional contaminants, subtle effect, limited data availability, and need to assess effects of land use and the climate change. So this is what I thought in the brief, just giving an overview at what challenges are today in the food safety assessment, risks, etc., which you need to have newer toxicological tools, but they should be the one which should be quite accurate, validated, and then used. Thank you very much.